The optimum pH for the activity of pepsin is about blank. To answer this question, we need to know what the enzyme actually is. Pepsin is a digestive enzyme that is located in the stomach, which is responsible for breaking down proteins. It is released after being triggered by gastrin, a hormone which activates pepsinogen, the inactive form or precursor of pepsin itself. Considering that this enzyme is found in the digestive system and is responsible for the breakdown of proteins, it is understandable that it would be located within the stomach. For an enzyme to optimally function in the stomach, it needs to favor the acidic environment which is provided due to the large amounts of hydrochloric acid. For this enzyme to function at its optimal efficiency, the pH level needs to be within the range of 1.5 to 2.0. Alongside the specific range of pH, the enzyme pepsin also needs to be within a specific range of temperature to operate well, which is between 37 to 42 degrees Celsius. Just like pepsin has its specific conditions that have to be met in order to function, there are many other enzymes that favor different levels of pH or temperature. As a comparison, the enzyme lipase prefers a pH of about 4.0, trypsin requires a level at around 7.6, amylase works best at a pH of 7.4, and maltase prefers a pH of 6.5.
Enzymes that catalyze the same reaction but have slightly different structures are called, the answer is, isoenzymes. The following choices would not be the best answer because they have different functions. For example, noncompetitive inhibitors have a structure that is different from the substrate. As it binds at the active site, it will distort the shape of the enzyme. A competitive inhibitor has the same structure as the substrate. It also competes with the substrate in order to reach the active site. Coenzymes and cofactors are the helpers of the enzyme. Coenzymes are organic materials that are necessary to adapt to the shape of the active site, which will allow the corresponding substrate to fit. Cofactors will also adapt the shape of the enzyme. However, they are composed of inorganic material that will increase the catalyst reaction.
A noncompetitive inhibitor has a structure that blanks. So let's begin by defining the definition of a noncompetitive inhibitor. A noncompetitive inhibitor is a molecule that binds to an enzyme somewhere other than the active site. Therefore, this rules out answers C and E. In addition, a noncompetitive inhibitor will change its shape so that a substrate cannot enter the active site and alter the 3D structure of the enzyme itself. Therefore, this rules out answers A and all the other options except for answer D. The answer to this question would be D. A noncompetitive inhibitor does not resemble the substrate structure. To summarize the answer for this question, in general, a noncompetitive inhibitor will have the following properties. The shape of the inhibitor will not have a similar shape to the substrate. It will bind away from the active site to change the shape of the enzyme so that the substrate cannot enter the active site. Also, adding more substrate does not reverse the effect of the inhibitor.